Greetings everyone! Today we will be explaining how central dogma works. But first, we need to know what central dogma is, how it works, and what does it do. After this video, you should know the following. First, you should know what central dogma is. Second, you should know the parts of it. Third, you should know how central dogma works. And fourth, you should know the functions of each part of the central dogma. So what exactly is central dogma? The central dogma of molecular biology is a key principle that illustrates the flow of genetic information within a biological system. It was first proposed by Francis Crick in 1957. The process begins with DNA, our genetic material, through a process called transcription. The information in DNA is copied into a molecule called messenger RNA. This mRNA then serves as a template for protein synthesis in a process called translation. In essence, the central dogma describes how the instructions in our DNA are converted into functional proteins, the workhorses of our cells. It's like a blueprint DNA being transcribed into work order mRNA, which is then used to construct a building protein.
Now, let's dive into the three parts of the central dogma. First, we have the process called transcription. What is transcription anyway? And why is transcription important in the process of the central dogma? Come with me and let's find out. Transcription is the process where the DNA sequence of a gene is copied into a molecule called messenger RNA. This is done by enzymes called RNA polymerases. Transcription is the first step in gene expression and is crucial to the central dogma because it allows the genetic information in the DNA to be transferred into a form that can be used to produce proteins. Transcription is essential to the central dogma of molecular biology because it's the first step in turning the genetic information stored in the DNA into functional proteins. During transcription, the DNA sequence of a gene is copied into the messenger RNA, also known as the mRNA. This mRNA molecule then carries this genetic information from the nucleus, where the DNA is housed to the cytoplasm of the cell. Here, in a process called translation, the mRNA serves as a template for building proteins. Without transcription, the genetic information in DNA would remain locked in the nucleus, unable to be used to create proteins that are vital for life. Therefore, transcription is a crucial process in the flow of genetic information from DNA to RNA to protein, as described by the central dogma.
Secondly, let's jump to translation, which is the second part of the central dogma. Now, you may ask, what is translation? And does it have any relation with transcription? Well, let's find out. First part, translation is the process where the sequence of a messenger RNA molecule is used to create a sequence of amino acids, which then form a protein. This process is carried out by structure in the cell called ribosomes. Second part, translation is directly related to transcription. The first step is the central dogma of molecular biology. Transcription creates the mRNA that serves as the template for translation. In other words, transcription and translation are two sequential processes that convert the genetic information in DNA into functional proteins. Without transcription, translation could not occur because it relies on the mRNA produced during transcription. So, they are interconnected steps in the process of gene expression.
Thirdly, we have what they call assembly, which is a key part of the central dogma. What is assembly, you may ask? Assembly often refers to the process of protein synthesis during the translation phase after the mRNA has been transcribed from the DNA during the transcription phase. It moves to ribosome for translation. Here, the sequence of mRNA decoded to specify the sequence of amino acid in a protein. The assembly part comes in when the amino acid are linked together in the order specified by the mRNA to form a polypeptide chain. This polypeptide chain then folds into a specific three-dimensional shape to become a functional protein. This process of protein assembly is crucial because proteins are the workhorses of the cell, carrying out most of the cell functions.
Finally, for the last part of central dogma, we have gene expression. Now, what is gene expression and what does it do? Well, there's only one way to find out. Let's jump to the final part. Gene expression is the process by which the information encoded in a gene is used to direct the production of a functional product, either a protein or a non-coding RNA. This process involves two main steps, transcription and translation, which are the core of the central dogma of molecular biology. During transcription, the DNA sequence of a gene is copied into messenger RNA. This mRNA then moves from the nucleus to the cytoplasm of the cell, where it serves as a template for the assembly of amino acids and into a protein during translation. The proteins produced play a crucial role in virtually all biological processes, making gene expression fundamental to all life. The regulation of gene expression allows cells to respond to changes in their environment, perform specific functions, and develop into different cell types.